C तो लिखना चाहिए था, तो ये होता है फिनोटाइप लिखना चाहिए, कैरियर और वो नहीं लिखना, मैंने भी नहीं लिखा, कॉम्बिनेशन में क्या है, देखो उसका वो स्पेशल फूड है, नमकीन फूड, बहुत सॉरी क्या, नमकीन में क्या, हाँ नमकीन में खाने के क्या है ना, ठीक है लड़की क्या क्या करना था, नहीं थी कि गर्लफ्रेंड में चैटिंग कर रहा था जो बनी नहीं है, अरे वो बनने भी नहीं वाली थी, वो रिजेक्ट हुआ है ना, वही जीत जाएगा मैं चला गुजरात और मुंबई में बहुत देरी है, मुंबई थर्ड, गुजरात ऊपर, थर्ड है, फोर्थ हो गई, अच्छा वो जीता था राजस्थान, गुजरात वालों ने चुनने की थी क्या शायद जीतने के बाद, आर वाज रेडी, अरे हां आर वाज रेडी हमें के गुजरात, हां बट रोहित शर्मा नहीं आएगा तो वही जीत सकते हैं, अरे अब बार बार थोड़ी ना सूर्य वहां पर डिपेंड है, आज फॉर्म चलाएगा उसका तो ए मादरचोद, कुछ भी और चाहे तेरे को आके पहले मैं कह रहा हूं ना रोहित का फॉर्म आना चाहिए है, और क्या वेंडन सूर्य कुमार ट्रांजिशन में है अभी, क्या होगा मिड कैंडिडेट और कैप्टन से अच्छा कर, कैप्टन भी अच्छा, वो तिलक क्लास में बॉलिंग पर अच्छा है, अरे सूर्य को हार गए ना भाई तू, अरे सूर्य को गया अभी आपने बोला है कम मिली पहले हार ना आज तो कम तो रात के 12 बजे सुनो वो तिलक अगर तिलक को क्लास में जो तिलक नहीं खेल रहा है, तिलक और मैं बोल रहा था ये नहीं आएगा, तू उठा दे से लेके आएगा ये, अगर टीवी हारे तो कुछ नहीं खेलेगा इसका, टीवी का वो आराम क्वालिफाइड है, वो हार भी गई ना कुछ प्रॉब्लम नहीं, बट मुंबई जीत तो बहुत हारने का नहीं सब कोड ही बहुत भाई अभी आएगा नहीं, अरे पर गुजरात वालों ने कुछ उम्मीद करनी चाहिए थी, पंजाब में उंगली की मार दी, आरसीबी ने भी उंगली वो बड़ा पाव बोल रहे थे, हां बड़ा पाव, अरे विराट कोहली वगैरह हाल करता मौका मौका खा रहे उसको, इसको अरे ये लोग वहां चिंता स्वामी में पड़ा रोज बड़ा पाव बोलते थे ना, कौन कौन लोग आरसीबी के फैन, तो मानखड़े में मुंबई के फैंस ने बोला मौका मौका, और लोग क्या मैच शुरू हो गया, मैं तो इंस्टाग्राम पे फाइट कर रहा था लोगों से कमेंट में ना, मैं देखता हूं हर किसी का, अरे रोहित शर्मा का सबसे ज्यादा रन बनाए नीचे लिखे, लेकिन विराट कोहली ने रोहित शर्मा से कम नहीं किया, मैं बोल रहा हूं तेरे को जस्टिफाई करना, वो वीडियो रोहित शर्मा के ऊपर बना है, अरे एक तो था पंजाब में, पंजाब का मैच जीतने का था ना, इकड़ा था मानखड़े में आके बुंड फाड़ दी तुम्हारी, पंजाब ने डाला, मैं तो यार पहिला बाजी में पंजाब में आके तुम्हारी बुंड फाड़ देंगे, मैच जीतने के बाद मैं गया उधर रो बाजी मार दी, भाई ये लोग वॉशरूम ही गए ना, मैं क्या कर रही हूं देखो ना जाके से एक साथ शादी के लिए सबको मारा अरे देखा पीछे मारा तो वही हुआ। अरे देखो मुझे की कलाई क्या भाई हड्डी हैंडी
यार पीछे छोड़ दे
मतलब उसके शॉट पीछे नहीं करते उसके शॉट ऐसे ऐसे सब पीछे भाई
ऐसा लगा
वो
सामने
आगे
से
से भाई उससे कुछ ज्यादा प्रैक्टिकल पीछे से मारता है ज्यादा प्रैक्टिकल हो गया
ये देखा है ये कर रहा है अरे बॉलिंग में अरे उसमें भी एक लास्ट ओवर का पूरा गाना मैच कर रहा था ठीक है दोनों दिल के ये क्या कर रहे हो रोहित का सिक्स
भाई जॉइन नहीं
क्या कर रहे हो बस वही कर रहे हैं आई फुल रेंज देखी मैम को लेके रेंज देखी मैम ही है मैम जल्दी आएगी 50 तो आएगी आगे से इंटेंट मार गया उसने गाड़ी पे ये हमने भाई डाटा वाला
आगे बढ़िया
कौन है भाई अगर थोड़ा ऊपर लगता ना कांच फोड़ के चला जाता
मैं तो सोच रहा था लगना चाहिए
वही ना मैं कांच फोड़ जाती देखने में मजा आता है
भाई उसमें कितना 5 लाख का नुकसान है
5 लाख का गाड़ी तोड़
मुझे भी 5 लाख का गाड़ी आ जाएगी
नहीं यार वो ईवी वाली 8 लाख की आती है
अच्छी आईपीएल का बजट
तो फॉर्च्यूनर अच्छा क्या ये डियागो दे रहे 5 लाख की को लगता है का बस पे हाँ मिलती है जो तीन देते उसे कैप्टन बोल कैप्टन का देने वाला पूरा
अब ये कौन रहा था इंडियन टीचर है ना
वो सूर्य कुमार गाने में बताने में भी बजा सुना ही नहीं
गाने में पहचाना नहीं होता एक हम उसके शॉट्स को निकालते हैं
हां रिवर्स शॉट I hate you.
उसका नहीं है
ये फर्स्ट क्लास में बैठी है तो भाई ये फोटो नहीं नहीं अबे वो ट्रेन यार में गाड़ी पड़ी रहेगी और चढ़ा के भीड़ बनी रहेगी
लाइट की वजह से
हां एक फोटो
सुबह के 5 बजे खाती होती है
इमेजिन
नहीं कोई नहीं उसके जगह यहाँ पर रोहित शर्मा भाई ऐसा हुआ ना रोहित वर्सेस और हम ये लोग स्टेरियोटाइप है कभी जीतेंगे नहीं भाई लास्ट आज भी एक भी नहीं जीते कब फाइनल बहुत बार फाइनल है सेमीफाइनल है बट जीते नहीं मुंबई फाइनल वाली बात है भाई मुंबई एक भी फाइनल नहीं आ रही है रोहित के अंडर
अरे आ गए
अरे हो गया तुम लोग का
क्या देखा है
हम नीचे देख रहे हैं वो नीचे छुपा दिया क्या छुपा दिया
जितेंद्र का इंस्टाग्राम अकाउंट देखे तुम
हां ये बेटे का दिया
80 के नीचे
बेटा होता तो समझ में आता
हां बेटी है
फालतू
बाल
अरे वो कभी मुर्गी सड़ा नहीं लगता ऐसे
वो ऐसे बात करता है,
दुनिया टेढ़ी
वो ऐसे करता है ऐसे।
और वो पूरी दुनिया टेढ़ा देखता है ऐसे लगता है
अरे ऐसे
वो देखने पास है टेढ़ा करके सबका शक्ल
उसका बाल भी ऐसा है ना,
उसके बाल वो क्लीन करते हैं।
अरे वो ऐसे करता है टेढ़ा
वॉशिंग में निकाला।
देखो नहीं खाली मार रही है
भाई हमारे सर ने वैसे करा। हाँ चल निकाल के खाते हैं।
उसने एक दिन बोला
ये
अरे ऐसे पढ़ाने में मजा लगेगा यार मैम
यहाँ से बड़ा क्लियर दिखता है, क्लियर।
भाई मेरे को डर लग रहा है टूट का अरे जितने देर घूमने गए
अरे
देख देख देख
तू ऐसे बोल रहा है, तेरा साला है वो, तेरे सर है।
डायनेस्टी
किधर गए डायनेस्टी रिजॉर्ट
अरे जितने भी लेट डायनेस्टी चल रहा है।
अभी कोई कौन है देख मैम क्या कुछ
अरे कोई नहीं है।
दरवाजा खुलने की आवाज
अरे इसका एक था ये कुछ तो
हो जाएगा देख हिंदी में मॉरल पढ़ा था जॉइन विद ब्रो जितेंद्र ब्रो जितेंद्र वो ब्रो
चक्कर
लक्ष्मण चट्टा
क्या क्या
मॉरल सिखाता है
भाई ये सर
उसके कमेंट सेक्शन में जय श्री राम
ए निकाल
लक्ष्मण चट्टा
उसके कमेंट सेक्शन
ए लक्ष्मण चाटुकार लक्ष्मण चाटुकार
चाटू जय श्री राम उसके कमेंट सेक्शन
क्या ही मारा चाटल है
तो चाटू हो गया ना चाटू चैटती का चाटू
ना कमेंट सेक्शन
अच्छा,
में
वो
जय श्री
अरे
राम
कमेंट शॉप है
अरे किड्स के लिए
उसके लेक्चर
मत उसका ईमेल
होगा होगा
अंदर दिखता है
चल बेटा
अगर मैम
डर रहा
भाई हो गया
किया तो काल का हो गया प्रज्ञान मजा
भाई भाई ये जन के बाल बिखरे हुए हैं।
ये दोनों ये जन को
आगे
बाहर लेके सर्च करते हैं कमेंट में जय श्री राम
हो गया टाइम का एक मिनट
ये भी टाइम कितना है।
अरे 1 मिनट 50 तक आ
चलो मैं आपको क्या
नहीं आई है
आपको क्या
गर्मी है तो मैं बंद है क्या
सबके सामने अरे भाई
ऐसे ही चिल
स्लीपर ट्रेन में बैठो ये स्लीपर बस में बैठो गाड़ी मैम देखो मैम को देखो
मुझे ऐसा भी है
तू भी ले लो
रिएक्शन
So now we were talking about imbibition. So the definition of imbibition, you will have to take a note. Take out your books, all of you. Take a note. Definition of imbibition. The definition of imbibition you are always asked in the defined section of the question. So see that you take that definition and remember a specific proper definition what to write when we are talking about imbibition. What did I tell you? What is the definition of imbibition to remember? What happened? Happened?
देखो नहीं तो तेरे पे हाथ किधर बैठा तू लाया वो देख
everybody हुआ था
जाओ उसके ऊपर कुछ रख दो नहीं तो the entire class will disturb like तो मधु को देखते देखते मेरा बाल
अरे
ऐसे
हाँ
जाएगा
बात है ना
हाँ
एक ही है
अबे how we done in
taking sort of
हाँ
nothing
कुछ करना है उसके साथ या तो मारो no problem I don't have any problem as long as that is harmful to you you can kill it there's no problem at all अगर उसको मारना है मारो उसको कोई चीज से ढक के रखना है ढक के रखो शरद will take care later but I'll give you just 1 minute to decide what you want to do with that after that कोई disturbance मुझे नहीं चाहिए
इसे रखे
देखो एक छोटा सा मधुमक्खी ऐसा huge big creatures
तुझे प्रॉब्लम
नहीं मुझे
अरे जाना टीम के लेके जा रहे हैं
प्रॉब्लम उसको ही है डर उसको ही लग रहा है
मुझे भी चाहिए
नहीं
मुझे भी चाहिए
इतना उड़ी नहीं लगा
उसके बाद कोई डिस्टर्बेंस नहीं करेगा so now we are talking about the process of inhibition so when I talk about inhibition remember certain concepts of inhibition to remember you are also asked about examples so examples we already talked about some few and the swelling up of the bark of the trees big plants basically we understand and that becomes a living example of course when we are talking about living and then what would be for the dead tissues we had already talked about the doors and the shelves and etc that becomes example for the living for the dead and this would be example for the living take the definition in your book the phenomena by which living or dead cells absorb water by surface attraction and examples you are supposed to understand so inhibition nothing much next we move on to a concept of diffusion we move on to a concept of diffusion so now when we are talking about diffusion what are we coming across what is diffusion what would be the definition of diffusion what would be the definition of diffusion who can tell me
the free movement of water
मधु तुमको डिस्टर्ब करेगा या तो उसको फेंक के आओ बाहर या तो कुछ करो
क्या दोस्त है
every time I turn back I should not see that something is going on in this line
ये बोल रहा है इधर
what is diffusion let us start with this now diffusion the second phenomenon that we talk about would be diffusion yes tell me the definition of diffusion what is diffusion
the movement of water molecules from a higher concentration to a lower concentration
no that is not the definition of diffusion when we talk about diffusion that can be applicable for any type of a molecule when we are talking about diffusion can be applicable for solids liquids and gases so what is the definition for diffusion to remember for diffusion always you should define this way that the process of movement of the molecules of a substance now that substance remember can be solids liquids or gases so when we are talking about
पेज दे
पूरी टीम लेके चल रहा
अरे बेचारा पेज टीम बस
आ जाओ सब पूरा बुकी ये ले उसके लिए भाई हिटमैन लगेगा
टीम वीकिल जब मैं अपने जब कोई गंदे नहीं करने दूसरी बार
आपने फिर से मारने फिर गंदा कर दिया
आगे ना आगे
आगे कौन नहीं किया
उसका नाम दे
ए इधर तेरा कैप पड़ा है
ले ना
उसके अंदर
अबे मेरा
ये ले इसके अंदर ही है
आधा तेरे ऊपर ही फिर
कहाँ पे है वो बताओ
डॉगी है क्या वो ऐसे कर रहा है देखो
टॉमी
मिंकू है उसका
बिल्कुल सही
मिंकू
सेवा डन नहीं कर तेरे को कंटिन्यू
वो तेरे को क्या बोलते कॉम्बो करेगा तेरे को पता है 7 नहीं 7 टेक्स कॉम्बो आगे जा
क्या हुआ तो आगे वो लोग है ना हम लोग काटेंगे
अरे तो आगे निकला
वो उधर
इधर से
ये सारे अपने
खाना नहीं है मूवियां नहीं
किसी का वायर है किसी का नहीं वायर
नहीं जिंदा है
अरे दिखा भी नहीं है
अरे उसको बोलो नहीं है
मैंने देखा है
नहीं मिल रही है
Now, what is diffusion? Let us now talk about this definition again. What is diffusion? The process of movement of molecules of a substance from its region of high concentration to a region of low concentration. So I say, when I talk about the process of diffusion, it can be applicable for solids, that means solute, remember. It can be applicable for any type of a solvent, and it can also be applicable for gases. Can we think of an example of diffusion referring to solids or solute? When I talk about diffusion of solids or solute, here we may understand the concept in all the three will remain exactly the same. Now I talk about a diffusion of solids, where I'm saying that I take a beaker, and in this beaker I take small amount of water. Diffusion of solute or solids I'm talking about. So what do I try to drop in here? I try to drop in maybe a cube of sugar. I try to drop in a small cube of sugar. Now the moment I take this cube of sugar, and the cube of sugar immediately settles down at the bottom. After that, in just a minute time, I take some water from here and I taste. Is the water sweet? Will the water be sweet? Will the water be sweet?
Yes.
No. Will the water be sweet? Not at all. The sugar cube I drop, in a fraction of a second it goes and settles down at the bottom of the container. I take small amount of water from here, a spoon of water from here, and I taste. Is the water sweet? No.
Till that time we stir it.
Even if we don't stir it, maybe after 5 minutes when I take the water from here and taste, now I get to know that sweetness. What has happened in this 5 minutes? The molecules of sugar, solute, the molecules of sugar had moved from a region of high concentration to a region of low concentration between the water molecules. And if I stir it, the process becomes even faster. Clear, no? So when I talk about, is the process of diffusion applicable for solids? Very much. Similarly now, I talk about again a tumbler of water or a beaker, and in this beaker again I take small amount of water, but to this water now what do I take is, I take a drop of ink now. So this drop of ink that I'm putting it here all settles down, or as it goes down through the water, this drop of ink that I'm putting here in the water, is it making the entire water color? No, it is not making the entire water color, only a small region of the water, only a small zone of the water is colored. So as the drop of ink passes, only a small zone is becoming colored. But after a period of 5 minutes, I get to see that the neighboring zones of the water, that is also getting that color, red or green or blue, whatever. So what has happened in this 3 minutes, 5 minutes, 6 minutes time? What has happened? The molecules of the ink or the dye has moved from a region of higher concentration to the surrounding areas between the water molecules. Yes or no? So can we talk about diffusion applicable for liquids? Very much. Can we talk about diffusion similarly? Can we talk about diffusion of gases? Can you give me an example? The concept remains exactly the same. Perfume spraying, correct. When we talk about the
exchange of gases like oxygen and
that is diffusion when you talk about the living cells. So, but like this, when I talk about an external media or I talk about a practical condition like this, now for this type of a condition, you could understand, yes, The burning of the incense stick, burning of paper, burning of anything, cooking, or the spraying of the perfume, etc. All these are good examples to understand, yes, the process of diffusion is very much happening. The process of diffusion is very much happening. So here again, when I say that I'm using a body spray or a perfume, the molecules surrounding me is concentrated. The molecules of the perfume are so very concentrated surrounding me now. So many molecules surrounding me. But a person sitting a bit close, a person sitting a bit away from me, can the person get the smell? Yes, the person can get the smell because the molecules are moving from a region of high concentration to a region of low concentration through the molecules of the air. The process of diffusion will always give an impression that it is applicable for all type of molecules, solute, solvent, gases, and all for a region of high concentration to low concentration. But now, are we talking about that the molecules of the ink and the water, are they in direct contact? Yes, they are. The molecule of the sugar and the water, are they in direct contact? Yes, they are. The molecules of the perfume and the molecules of the air, are they in close contact? Yes, they are. So what is common in all the three? For the process of diffusion to happen, the two category of molecules that we are talking about should be in close contact. Are we talking about the movement of one molecule from one region to another crossing a membrane? No, there is no membrane crossing as such. And the moment we talk about membrane crossing, we are talking about the concept of osmosis. The moment we are talking about membrane crossing, we are not talking about the movement of gases and solute. The moment we talk about membrane crossing, it would be only and only for solvent, liquids. Only for solvent and liquids. So now, if that is diffusion that we had already talked about, now we try understanding about osmosis. Then what is osmosis? How is osmosis different from diffusion? Osmosis is the process in which molecules of solvent will move from a region of high concentration to a region of low concentration, passing through a semi-permeable membrane. Correct? So what is the difference that we have been talking about diffusion and osmosis? In the case of diffusion, molecules are moving from a region of high concentration to low concentration when the molecules are in contact. The molecules have to be in contact. The two types of molecules that we are talking about, diffusion, the molecules are in contact, but there is no passing through a membrane as such. But whenever I have to talk about osmosis, first thing is, I cannot talk about solute. I cannot talk about molecules of gases. No, osmosis is only for the movement of the solvent molecules, water molecules. But something that is common is from a region of its high to a region of its low, that remains the same. Let us start writing one answer there. Write two differences between, put the name of the chapter, write two differences between diffusion and osmosis. You will be getting questions as per conditions in the bracket. So I give you two conditions. Two differences based on the condition given in the question paper, you will have to write down the point. Two differences, diffusion and osmosis. The first point, make a table, diffusion and osmosis. Make a table, diffusion and osmosis. Write down. Under diffusion, the movement of or the phenomena in which molecules of a substance, phenomena in which molecules of a substance move from its region of high concentration to its region of low concentration, when The molecules are, when the molecules are in direct contact. When the molecules are in direct contact. Other side, osmosis. Movement of molecules of solvent. Movement of molecules of solvent. Movement of molecules of solvent from a region of high concentration to a region of low concentration to a region of low concentration through a semi-permeable membrane. Through a semi-permeable membrane. Through a semi-permeable membrane. Next point. Diffusion. Applicable for diffusion. Applicable for solute. Applicable for solute, solvent, and gaseous molecules. Applicable for solute, solvent, and gaseous molecules. Applicable for solute, solvent, and gaseous molecules. Other side, for osmosis. Applicable for molecules of solvent or water. Applicable for molecules of solvent or water. Applicable for molecules of solvent or water. Now we try understanding a concept of osmosis. We try understanding a concept of osmosis. What is this that I'm trying to show you? I am talking about two types of molecules here, water and solute. Okay, two types of molecules. And what is this membrane that I'm showing you here? This is a semi-permeable membrane. This is a semi-permeable membrane. Now here let us try understanding. In this chapter, you are going to come across various examples of semi-permeable membrane. Now, what is the definition of a semi-permeable membrane? That also you have to understand. What is a semi-permeable membrane? What is a semi-permeable membrane? A membrane that will allow the movement of the solvent and will not allow the movement of solute. A membrane which will allow only the movement of the solvent but not the movement of the solute. That is a semi-permeable. So that's the reason whenever we talk about the cell membrane, we say cell membrane is semi-permeable. Then what is the property immediately you should remember? The cell membrane will allow the movement of the water and not the movement of the solutes. The solutes, but then we know that the solutes also move through the cell wall and the cell membrane, but that has to be in a different process. For that, a different process altogether. But if I have to talk about osmosis, no. Osmosis, only the movement of water molecules. So here what I am trying to show you is that this is a semi-permeable membrane, and just now I told you what are the properties of the semi-permeable membrane. It will allow only the water to pass through, not solute. What are some examples of the semi-permeable membrane? Just shortly we are going to come across some of those experiments now. What are some of the examples of the semi-permeable membrane? Cellophane paper. Cellophane is one. Then we talk about the parchment paper. We talk about the parchment paper. Then we talk about the animal bladder. These are some examples you will come across. Animal bladder or the goat bladder also it is asked. It is mentioned in the paper. Then we talk about the egg membranes. The egg membrane. These are all semi-permeable membrane. Why? Because all of them are known to have more or less a similar properties. They will all allow the movement of the water, but hold on the movement of solutes. It will not allow the solute molecules to cross. So these are some examples to remember. Now here we are trying to understand two different media on either side of the semi-permeable membrane. Very simple. What am I showing you here? Where is the water molecule more? This side. Solute molecules more this side. So if this is a condition, what should be the direction of the movement of the water? The direction of the movement of the water would be this way. From a region of its high to a region of its low. But can I easily explain you this also, that the movement of the solute would be this way? Not exactly. For the movement of the solute, a different process altogether is there. So you are not to believe exactly this way, that if the water is moving this way, then the solute also must be moving this way. The solute molecule movement again has got a little bit of a different method process, not exactly like the water. So this is not there. So I'm talking about that the water is always moving from its region of high, to its region of low. I talk about a plant cell now. I talk about a plant cell, and as usual, this particular plant cell should be having the cell wall and the cell membrane and the vacuole. Now, when I talk about that this particular cell is taking in the water molecule. So what is the process we are learning? We are learning a process known as osmosis. And we are saying that if it is osmosis, we understand that surrounding the cell, there is a whole lot of water molecule. And comparatively inside the cell, in the vacuole, the amount of the water molecule is less. So if this is a situation, then the cell will constantly take in water, and that also not in the space between the cell wall and the cell membrane, but directly into the vacuole. The pressure, the osmotic pressure is much more than the imbibitional pressure. So the water molecule is constantly entering and becoming a component of the sap. When the cell is absorbing water, taking in water inside, what do we call this as? We will call this as endoosmosis. So for endoosmosis, when will endoosmosis happen? When will endoosmosis happen? When the outside environment is hypotonic and inside the cell, the environment is hypertonic. When will the cell show endoosmosis? When surrounding the cell, there is so much of water. Surrounding the cell, it is a dilute environment. Surrounding the cell, it is dilute. Then only the water will enter. Endoosmosis. And with regular endoosmosis, we may also come across that the cells may also suffer from one condition. The cells may also undergo a process known as swelling up or bursting. The cell may also undergo a process known as swelling up or bursting. So now try to understand this type of a situation. To understand this, let us come across certain conditions first of all. So if that is so, we are to come across under what different condition osmosis happens. Okay, now here I am just talking about 3 different beakers, and in each of these beakers, try understanding these terms. Very simple. Once you understand, there's nothing to read and read and read. So here what I'm saying is, I talk about 3 different beakers. In each of these beakers, I have taken some, or I've been given some solution. There's a solution. So in each of these beakers, there is a solution. I don't know what is the strength of the solution. When I say strength of the solution, what are you supposed to understand? Strength means how much of the solute is there in the solvent. All these have same amount of solvent. But how much of the solute is added? Solute means it can be sugar or salt, for an example. How much of the sugar or salt is added, that we don't know. So that's why I'm saying I don't know what is the strength of the solution. What is the strength of the solution is unknown. But yes, solvent is the same amount. The water part is the same amount. Liquid part is same. Then what do we do? Some cells are taken here. Let us take some plant cells only. So we are taking some plant cells. So what has happened? We take some plant cells inside. So some plant cells are taken inside. Since we are talking about plant cells, let us give an example of plant cells, but it can be applicable for animal cells also. So now some plant cells are taken inside. So what is it? Some plant cells are taken inside. What about the third? Some plant cells are taken inside. So now we try understanding that after we take these plant cells and the solution, whatever strength the solution is, we wait for a particular period of time. We are asked to wait for a particular period of time, maybe for 8 to 10 hours, 12 hours, we wait. After that, we get to see the behavior of all these cells. Now that is what we are supposed to understand. In the first condition, we get to see that the cell shape remains almost the same. Whatever the cell shape was there, it remains exactly the same. The cell shape remains exactly the same. What about the second? In case of the second, we get to see that the cell has undergone swelling. The cell became swollen. Remember, this is applicable both for plant and animal cells. Again, I repeat. So the cell became swollen. What about the third condition? In case of the third, the cell became shrunken. The cell became shrunken. The cell has undergone shrinkage. What has happened in all the three conditions? In one, the cell remained as it is. In the second, the cell underwent swelling. And in the third, the cell became shrunken. Now, we have to understand that what must have happened in each of these cells that has given three different results altogether. If I talk about the first, how is it possible that the cell must have remained exactly the same? The cell will remain exactly the same only when the amount of water molecule entering and the amount of water molecule coming out is almost the same. If in a molecule, if in a cell, the amount of water entering, I talk about the same cell again here. If in this cell, the amount of water entering and the amount of water coming out is almost the same, then the shape of the cell will not show much of a difference. But the question is, when will this happen? Under what condition will this happen? When the net gain of the cell is almost zero. The net gain is zero. The cell is not preferably gaining anything much, because if it is gaining 6 molecules, it is giving away 4. If it is getting 3 molecules, it is giving away 2. So ultimately gain is nothing much. So that's the reason the cell is not showing any change. When will this happen? It will happen only when the outside of the cell and the inside of the cell has the same concentration of solute. When the outside of the cell and the inside of the cell has the same concentration of solute, so the water molecule entering and leaving will be more or less the same. So under such type of a condition, what will we name this solution as? We are naming the solutions here now, based on this movement. So what will be such type of a solution known as? Such type of a solution will be termed as isotonic. What is the meaning of isotonic solution? A solution which has the same concentration or equal concentration of solute as compared to the cell. A solution which has equal concentration of solute, or I may say water also, no? यहाँ पे water और solute लगभग same है cell के जैसे. That is the reason the cell is not needing, the cell does not need to take also any, or is not in a situation to give also much. Under such situation, the solution that the cell is in, we will say the solution is isotonic. We'll have to learn all the definitions. Hello. I know this chapter is going to be a little bit draggy, but you have to keep yourself awake. Otherwise, I'll also go down the last bench and I'll also sleep. Jago. Now, isotonic. ये absorption और transpiration ऐसे ही जाएगा भाई, इसका कुछ नहीं कर सकते, ऐसे ही जाना जाएगा. It's not like other chapters now that we talk about the circulatory, endocrine, and excretory. That's becoming very physiological chapters these are. But this you will get 10 mark questions from this chapter. 2 से 3 question तो आना ही है. So you have to know this concept. So isotonic, what I told you, what is isotonic? ये जब भी सोएगा ना इसको थोड़ा धक्का मार. हाँ? और नहीं तो वो मधु तो है ही वहाँ पे. उसको सामने थोड़ा लेके आने को वो आराम से जग जाए. So now that we talk about isotonic, what did I tell you? What is the meaning of isotonic solution? A solution which has equal concentration of solute and solvent as compared to a cell. Now remember, we are actually talking about a concept of tonicity also here, because here we are also trying to understand that why should the water molecule move? Where will the water move? Why should the water move? Before I talk about the concept of osmosis. So here, what we are saying that the water will always move from a region of high to low, but here to high low kuch ho hi nahi raha, because yahan pe the water is almost the same inside and the solutes are almost the same as inside. So we are comparing, remember, we are comparing. The moment we talk about this type of a concept, we are actually comparing two different. Talk about the second condition. In the second condition, what is happening? In the second condition, what is happening? If this is the plant cell and this is the vacuole, what is happening in the second condition? Why did the cell swell up? Because the cell has gained so much of water. If it has gained 10 molecules, then hardly it must have given out 1. If it has gained 10 molecules, then hardly it must have given out 2. 15 molecules, 20 molecules, hardly 1 or 2 molecules. So it has gained too much. Then what it gave out? So the cell became swollen. What is this process known as? The process of movement of the water will be termed as endoosmosis. Endoosmosis. What is endoosmosis? Movement of the water inside the cell. When the cell is absorbing water, when the cell is taking in water, endoosmosis. And when will endoosmosis happen? When the cell is in what type of a solution? When the cell is in a dilute medium. When the medium outside the cell is dilute, when the medium outside the cell has got more of water, then only the cell is absorbing water. So what type of a solution we will call this as? This type of a solution, we will have to name it as hypotonic. Hypotonic. Now what is the definition? How does the definition go? Hypotonic solution, a solution which has got higher concentration of water and lesser concentration of solute as compared to a cell. Again, it is relative, huh? Remember, the moment I'm talking about all these concepts, these are relative. मतलब एक के साथ दूसरे को compare करके ही तुम बता सकते हो. I cannot only say like this, that this is a solution, hypotonic solution is one which has got higher concentration of water and less concentration of solute. It is incomplete. Higher, matlab the moment you say higher, it has to be compared with something, no? Higher with what? With a cell. Come to the second, third. Now you should be able to tell me something. When I say that the cell is getting shrunken, what must have happened? If this is the cell, what must have happened to the cell if it is getting shrunken?
The water must have moved out of
The water has moved out so much as compared to what it must have got. It must have lost 10 molecules, and in return, by chance, it must have got 1 or 2 molecules. So the giving out is much bigger than what the cell got. But remember again, I cannot tell you straight away that the cell is only losing water and the cell is not gaining anything. No, the cell would gain also, but the loss is more. The loss is more, the giving away is more. If this is endoosmosis, then what should we talk about this as? This is exosmosis. And what will we name this solution as? What is this solution which has got a higher concentration of solute, less amount of water? That's the reason the cell is giving away water. No? This type of a solution, we will name it as hypertonic. Hypertonic. So what did we learn? 3 types of solutions we are coming across now. And these 3 types of solutions, what are these? Isotonic, where we are talking about the concentration of the water and the solute as compared to the cell is same. Hypotonic, when I compare a cell, hypotonic, the cell is in hypotonic medium. Water is more outside or solute is more outside? Water. The cell is in hypertonic medium. Water is more outside or solute is more outside? The cell is in hypertonic medium. Solute is more or water? Solute is more. The outside medium is concentrated. Aaya ye samajh mein? Now, if this is clear, then we move on to the concept. This was actually a concept of tonicity, but I took it earlier so that you understand what is endoosmosis, what is exoosmosis, and the concepts about osmosis now in the form of certain activities we understand. The same concepts and the rule of osmosis now try to follow, and we will be talking about certain activities here. In this chapter, there are a whole lot of activities that you should remember. We are talking about certain setup, and in this setup, we talk about the presence, or we take a beaker, and this beaker is filled with almost half filled with water. And what do we take is a thistle funnel. We take a thistle funnel. This is called a thistle funnel. And inside this thistle funnel, what are we taking? What kya ho gaya Raj? Neend aa raha hai? Huh? Absorption ye hai. Abhi tum logo ko neend dega. You want to go out for 2 minutes? Fresh up. Eyes wash karna hai. मुझे लगा तुम लिख रहे हो. Listen carefully now. So now when we are talking about osmosis, सब लोग Chinese बन गए. So now when we are talking about osmosis, what is the setup? Listen carefully. After that we'll do some questions. When we are talking about osmosis, what is there in the beaker? Water. What is there in the funnel? A particular type of a solution. We are not talking about what is the strength of the solution. Nothing. And we put the thistle funnel in such a way that we invertedly place it on the bottom of the beaker, and the mouth of the funnel has to be fastened, tied with a semi-permeable membrane. And I already gave you some names of the semi-permeable membrane. Now we talk about cellophane. So this is the setup. In this setup, we keep this setup under normal room temperature and wait for certain few hours. After certain few hours, what are we supposed to observe? After certain few hours, what are we supposed to observe? So this is the first activity that we are talking about. And in this first activity, what did I tell you? It is a comparison between the water and a particular type of a solution, remember. So what are we supposed to understand? This is a medium which is hypertonic because I am talking about a solution. Yes or no? This is a hypertonic medium. When I talk about solution, you always understand solution means there is water and some amount of solute is there. Even if 2 crystals of sugar, then also some solute is there. What about this? No, we are not talking about any solute here. So which of these environment, this or this, becomes a hypertonic environment? This one is hypertonic. This one is hypotonic. ये हो गया hypertonic. ये हो गया hypotonic. Then tell me, from where to where should the water molecule move?
Inwards.
रात में तो सोएगा. हाँ बोलो.
From high concentration to
कहाँ से कहाँ? From where to where? I'm talking about 2 medium here. There's a beaker, there's a funnel. From where to where should the water molecule move? From the beaker to the funnel or from the funnel to the beaker? The beaker to the funnel. So we understand that the movement of the water molecule will be definitely this way. Why? Because the movement of the water will always be from a region of high concentration to a region of low concentration. After a particular period of time, then what are we supposed to observe? After a particular period of time, we will observe if this was the initial level of the solution. Then after a particular period of time, we are going to observe that the solution level will increase. So we understand that this is a changed level of the solution now. What will be the strength of the solution? Increasing or decreasing? Increasing or decreasing? समझ के बोलो. Strength मतलब जो concentration था, that concentration will even become more or lessen? Less. Concentration will lessen. Will become less. I talk about some 1 teaspoon of sugar added. So now after 6 hours or 10 hours, the sweetness will be more or less? Less. Because so much of water has been added. So now I understand that the concentration has dropped. Less ho gaya na concentration. So this is one experiment that you are supposed to understand. Now what I'm doing is, now I'm taking a 20% of a solution. That's the second activity. Now I take 20% of solution here. I take a 20% of sugar solution here. And same water outside. Wait for a particular period of time. And then observe the change after a period of some 8 to 10 hours. So initially I said the solution was with just few crystals of sugar. But now I'm saying 20%, meaning 20% solution means 80 parts water and 20 parts कोई solute है. Now what will be the change after a particular period of time? अब क्या change होगा? ये water same है हाँ. यहाँ पे there is no change I am talking about. I am only talking about the change in the concentration here. पहले कम था, अभी ज़्यादा है.
So maybe it will be funnel even deeper because there is 80% of water, so there is more water compared to
80 parts of water I said. Percent नहीं बोला मैंने. I said 80 parts of. when I say 20% solution मतलब 20 parts कोई ना कोई solute है and 80 parts water है. Understand ना? Earlier water was there and a dilute solution. I said just few pinches of sugar I told you, no? And a dilute solution. Now I'm saying 20% solution मतलब ये solution now that I'm taking is a more concentrated solution than before. But outside same water. Change observation after 8 hours. Think. Rule याद रखना है. Osmosis. What is the rule? Why?
Because in the solution it has got 20 parts of solute, but in the beaker it's only water.
0. Correct.
And the level of water will drop.
Very good. So the same concept. Whether I take water and 1% solution, remember, water and 15% solution, water and 20% solution, water and 30% solution, ये तो फिर भी hypotonic ही हुआ ना? This is still hypotonic. The moment I take water and 1% solution, also if I take that 1% solution is a hypertonic medium. And the water will always move from hypo to hyper, dilute to concentrate. So now I'm saying 20%. So obviously this will be the direction of movement of the water. Is this clear or no? Clear? Sure? Are you sure? Not clear? When I'm saying this is water and this is a concentrated medium, this is water and this is a concentrated medium. Let that concentration be 15% or 1% or 2% or 20%, whatever. The moment I say 1%, 2%, 5%, so you understand कुछ ना कुछ solute तो है. तो अगर solute है, now that the solute is there, and यहाँ पे तो कोई solute है ही नहीं. So which is a hypotonic medium? ये हो गया hypotonic, only water. ये हो गया dilute medium. And the rule of osmosis says that the water will always move from a region of high to a region of low. So water should always move from here to here, here to here, here to here. यहाँ से यहाँ move होने का तो कोई सवाल ही नहीं है. So now that we are talking about that the water is moving from here to here, then what are some of the differences? The water level will drop here in the beaker and the solution level will increase in the funnel. Am I clear? आया समझ में? So this is the first concept that we talk about. What is the next now? Now with the same concept, again I am talking about a solution hai, any solution. Always remember, the moment you are talking about a solution, this becomes hypertonic, this is hypotonic. Unless and until I talk about a solution here also. I'm still talking about water, I'm still talking about solution. So jo bhi hone do, solution, whatever the strength of the solution, you are understanding solution means it becomes a hypertonic medium. This is a hypotonic medium. Now I'm saying that now we are not using cellophane. Condition यहाँ पे same है, solution and water. Condition same है. But now I'm not using cellophane, rather I'm using a rubber sheet. I'm using a rubber sheet or a plastic sheet to fasten the mouth of the thistle funnel. So this cellophane and rubber sheet, how are they different? The cellophane allows the movement of solvent but no solute. The rubber sheet will not allow the movement of the solvent also and the solute also, because it is impermeable substance. It is impermeable absolutely. What is impermeable? Does not allow the movement of anything. Solvent, solute, nothing. Plastic, no plastic when we talk about. The plastic sheet, the rubber sheet does not allow the movement of solvent, solute, anything. Then if this is the small change that I make in this activity, then what should be the observation after a period of some 8 to 10 hours? What do you think? Level of water ये है, level of solution ये है, and here it is a rubber sheet that I take now.
No change. Now it will remain the same.
The level of the water in the beaker and the level of what will happen to the level of the solution in the funnel?
Remains
It remains same.
It will remain the same.
because this is an impermeable.
Since it is impermeable, it will not allow the movement. Even if there is a difference in the concentration, even if there is a difference in the concentration, this is not allowing the movement possible. It is not allowing the movement possible. But had this been cellophane, then what? This way movement, beaker to funnel. And this movement from beaker to funnel, till when will it happen? As long as the concentration here and here is the same, equilibrium reached. So experimentally that is very difficult to reach that particular type of a condition. But yes, that's a concept to understand that how long will the water move had this been cellophane. How long will the water move? The water will move as long as on either side of the now this is a semi-permeable membrane, on either side of the semi-permeable membrane, unless and until equilibrium is achieved. Till that time, the water molecule will keep moving. Okay, now the setup remains exactly same. Now I'm not using rubber sheet, but I am using muslin cloth or a muslin sheet. I use muslin cloth. What is muslin cloth? Muslin cloth means those fine cotton cloth that we have where the pore size is big enough. The pore size is quite big enough, and which will not also hold the solute molecules. It will allow the water as well as the solute all to run through. Which will allow the water as well as the solute all to run through. So if that is the case, then what should be the change? अब cellophane भी नहीं लिया, rubber sheet भी नहीं लिया, but I am taking a muslin cloth now. Gargi, what should be the change you feel? यहाँ पे अभी muslin cloth is there. Otherwise, everything is same. Think again. I told you already the condition. If it is a muslin cloth, it is same like the cloth, cotton cloth, where the pore size is quite big enough, and I fasten it here. What should happen? Sorry, huh?
Beaker level will increase.
Water in the beaker will increase. Why? Why?
Because it is permeable.
Because it is weight.
Permeable.
Because it is
Permeable.
freely permeable. The muslin cloth is freely permeable. Muslin cloth is freely permeable. Same like the cell. Same like the cell. The muslin cloth is freely permeable. So now the direction of the movement of the water is not going to be this way. The direction of the movement of the water will be this way. The direction of movement of the water is going to be this way. It will not be this way anymore. Q. Irrespective of the concentration differences. Now we are not talking about only the movement of the water because this muslin can allow the solid also to run through. Even though there is a huge amount of concentration difference, this is dilute and this is concentrated, but it is not allowing the molecules of the solid to hold back. So all will run out. And because of that, the level of the water in the beaker will increase. And if the solute is sweet or sugar, then this water will gradually become sweet. And the level of the solution will gradually fall. The level of the solution will gradually fall. Yes or no? So these are the three conditions that you are supposed to understand. One, we have talked about the typical condition of cellophane. Another, we have talked about the freely permeable, and another condition was with impermeable. So three basic conditions we have learned. Let us solve one or two questions based on this concept. Then we move on. See in your textbook, there are some specific questions based on this. Some specific, there's one more whisking bag that also we'll have to look into. But before that, let us go to certain questions. Turn to page number 53. Turn to page number 53. Try number 2. Try number 2. Try number 3. Try number 2. Try number 3. No, E. E that is, no? E is the diagram base. Your short answers will be at the end of the chapter. Number 2 and 3. Go through the question, see if you have understood the concept. For the rest of you, 3.15. 3.15, question number 13. 3.15, question number 13. Next is already over in school, no? No, not a problem. Just go through. If you have any doubt, you can tell us. And 3.17, question number 25. 3.17, question number 25. These two questions you all will do. Got those two questions that I told you?
Yes.
Whatever. See if you have understood this concept. Those of you who have finished the previous questions, you may try this one. If you have finished the previous two that I told you already, if you have no doubt, then you may take this question. And you all will finish the two text questions that I asked you, then you will go through this. Any doubt? You will?
Question 2, G. And G.
Mention 2 advantages of the process, 2 plants. Osmosis. Mention 2 advantages of the process, 2 plants. 2 advantages, transportation, photosynthesis. Mention 2 advantages. We are talking about osmosis. So the process is all about osmosis. What are some of the advantages that the plant enjoys for the process of osmosis that it shows? Osmosis, because of osmosis, it takes in water, which helps in the process of transportation of food and minerals. Because osmosis, the water can move up, the water minerals can move up to reach the leaves of the plants and carry out the process of photosynthesis. So transpiration, transportation, photosynthesis, these are some of the advantages that we are supposed to have. You may just write down the 2 specific words also, then also it's okay for you. Transportation, photosynthesis, transpiration, advantages. These are the benefits the plant enjoys.
What do you observe in the experimental setup after an hour or so?
Which one?
13.
Which one?
It is not mentioned, it is permanently or semi-permanent.
Which one? Vital process. Study the same and okay. In the process, depend. Let's see the rest of the questions. In the experiment after an hour. What control? Keeping in mind the root hair cell and its surrounding. So name the parts that corresponds. So we are talking about a root hair cell. So if it is a root hair cell, then obviously we are talking about a cellophane membrane here. Or a semi-permeable, whatever, semi-permeable. So based on that only a answer. Now, before you start answering all these questions, there is something more that we are going to talk about. That would be a concept of control experiment. We have been talking about all these. What is a control experiment? What is a control setup? A control setup would be such where at the end of the experiment or at the end of the time, you are not going to see any observation. There will be no observation, and that's the reason we can compare with our other setup. So if I have to talk about a control setup, then what should I do with this? This is one of my experimental setup. I have to parallelly talk about a control setup. Then what should my control setup be? I take a beaker, and if this is the funnel that I'm taking, then the cellophane is there. And if I have to talk about a control setup, can you tell me? I just now told you, if it is a control setup, then at the end of the experimental time, I don't get to see any observation. So to say that, I may take water here and water here also. I take water here and water here also. Then am I going to see any change after 8 hours? No. Or I may take the same strength of the solution here and here. That also I can do. But that is a bit difficult. I may take water here and here. So that would be a control setup. So what is a control setup? Which does not show any observation and used always for a comparison. Always used for a comparison. So we may come across such questions. Control. If you have finished the text questions, then try this one. You may not write it, just go through the question and see if you know the answers. If you know the answers, good enough. If you don't know, ask. Gargi, have a doubt?
Not in the parchment paper.
Parchment paper.
Parchment paper also acts like the
Cellophane, semi-permeable. This question, no, the first one. You are basically supposed to write osmosis only. Don't write endo or exo. Could you solve the 2 questions?
I mean, this.
First one.
Karo fast.
I mean, this the third question is reason for all the observations.
What
So
is
we
the
just-
reason for the observation?
So when just we mention the OSCE, so you have to explain that.
What is the reason? What happens to the level of the liquid in the funnel?
I mean, it increases.
It will increase. What is the reason?
Uh,
Water moves from the beaker. The water is moving from the beaker to the funnel. Because outside the funnel or the water in the beaker is a hypotonic medium. And this is a hypertonic medium. Water moves from a region of, the reason is water moves from a dilute to a concentrated medium. Those of you who are solving the other questions, take a look into this question and see if you know the answers for all. If you don't, ask. The moment I give you questions to do, all of you start talking. You should immediately start looking into the question and solve fast. Not look at me, look into the question and find out if you know the answer. So all of them. I'll give you one more. पहले ये complete करो.
How can we justify the parchment paper selectively coordinated?
Because the parchment paper is allowing only the solvent molecules to pass. It is not allowing the solute molecules to pass.
Justification which you also impress the level of water in the funnel is increasing.
So the level of the water in the funnel is increasing only because the water is moving through this parchment paper, which is allowing only the solvent molecules to pass and not the solute. Done? All of you have gone through these questions?
Yes.
All of you check here, all of you take a minute, look here. What happens to the level of the water in the beaker after the experimental period of time?
Decreases. What is the bigger decreases?
What is happening here? We are talking about that this is water and this is a solution. This is sugar solution, this is water. What happens to the level of the water in the beaker? It will decrease. What happens to the level of the liquid in the funnel? Increases. Reason for observation, water moves from the beaker to the funnel from a region of high to a region of low concentration or from a dilute to a concentrated medium. Any of these you can write, all are correct. What is the reason? Okay, will there be any change in the taste of the water in the beaker? No, because the solution is not moving this side. The water is moving here. This is progressively becoming dilute. This will not become concentrated anyhow. Will there be any change in the taste? Okay, after the experiment, how can you justify that the parchment is selectively permeable? The parchment is not allowed the solute. The parchment is not allowing the solute to come down, no? Because if the parchment would have allowed the solute to come down, then this water would have been sweet after some time. But this never became sweet. But this kept on increasing. So we are very much clear that the parchment did not allow the movement of the solute, but only the movement of the water. All clear? Go through this set of questions. Almost the same, but little bit of a difference in the concepts. Raj, go through the questions also. Clear? Name a semi-permeable membrane. Okay. What happens to the? I gave you 4 examples already. What happens to the level of the liquid in the funnel? Liquid in the funnel.
Increases.
Here it is water, here it is beaker. What happens to the level? It is increases. If the beaker contains hypotonic, here, increase. What is the reason? Same like before. Will there be any change in the taste of the water of the beaker? No. When will osmosis stop? When
it means
concentration on either side of the membrane, this and this. These are the two different sides of the membrane. This is the cellophane, one and the other. On either side of the membrane, when the concentration is same, equilibrium is reached, then only the movement of water molecule will stop. Define semi-permeable. I already told you what to write. Semi-permeable definition, what to write. A membrane which allows the movement of the solvent molecules and not the solute molecules at all. What is the next questions fast now? Then we move on to one more concept.
Now, please show the PSS specific.
These questions now, those of you are using Selena, I always repeat, be very good friends with this four, and take images of the questions that they have in their book. And if you're not good friends with them, they will not give you. Don't give them. Give them misbehavior. Don't give them. So the questions in this book, there are many questions which you will as it is get to see in the paper. Selena, roughly questions are comparatively lesser, but the contents are exactly up to the syllabus. So that's why we follow Selena. Exactly what is there in the syllabus, Selena has all of it. But this book, the other book here, which has got so many extra concepts which are actually not necessary for you. So when those of you are reading the other book, see that you know the syllabus nicely. Otherwise, you'll end up reading unnecessary topics, also waste your time. Reading and knowing, not a harm at all. But if you can manage, that's very good. But see that in that process of knowing, reading, you don't leave up topics which are in the syllabus and you don't focus on them properly. Done. Now, let us talk about one more concept. Text questions all over. Anybody has a doubt? All over? Both questions, what the answer? Any doubt that side? Text questions?
I mean, control setup, don't have to draw, we have to explain.
What is the question they are asking you?
What control setup can be set up for the
What control setup can be taken? I told you, water both in the beaker as well as the funnel.
No, she's asking that we have to draw it and show.
Are they asking you to draw? No, I think no. For control setup, they will only ask you to mention, not draw.
Ma'am, this mention two advantages of this process to the plant.
So I asked, he asked already. What are the advantages? I had mentioned 4 advantages that the plant enjoys. Transportation, photosynthesis, transpiration, photosynthesis, transportation. All the 4. Mechanical stiffness.
Same for also diffusion.
Same for also.
Question answers what are the advantages of diffusion for plant?
What are the advantages for diffusion for a plant? Diffusion, not exactly. Diffusion advantages would be photosynthesis because carbon dioxide diffusion we are talking about. That is one. But no, we cannot talk about transpiration that way. Transpiration majorly we understand roughly also because the water molecules, the water in the form of vapor gets diffused roughly. Only this one. Mechanical stiffness is all these things are not applicable at all when you talk about diffusion. What else here we are talking? Transportation is not allowed. Transportation will not be. Photosynthesis with which you can write. Photosynthesis, of course. Diffusion of carbon dioxide. Transpiration, of course, yes. Diffusion of water vapor. Now we talk about the same concept here again with another type of a semi-permeable membrane known as the whisking bag. The whisking membranes, the whisking bag. Now the features of the whisking bag and the thistle funnel that we are talking about almost the same thing. We have a beaker here. Check here what's happening. We talk about a beaker. This beaker has got water. And we are talking about a whisking bag. So quite often what is also done is some some glass tubes are also added, some glass tubes are also inserted. So here one glass tube is also inserted quite a number of times. Let me see if I can show you that. See here, so when we are talking about this whisking bag, just try understand that this is a whisking bag where inside this whisking bag there is a solution. Outside the whisking bag there is just water. So we are taking a beaker. In the beaker there is water, same like the previous. And we are taking a whisking bag. The membrane of the whisking bag same like cellophane, with little bit of mesh size change. Otherwise it is same. And this whisking bag we have taken a solution. We wait for a particular period of time. After a particular period of time, what are we observing is, after a particular period of time, we are observing that this whisking bag is taking water from the surrounding and it is constantly getting swollen. The water is getting filled up inside the whisking bag. But the level of the water is never rising, meaning the solution from the whisking bag, the solution also has got some amount of water. That water is never coming out, rather the water is getting in. So comparatively, if I think of this whisking bag as a cell, then what type of osmosis is happening here? If I consider this as a cell and the whisking bag is gathering water and becoming swollen, what type of osmosis I'm talking about? Endoosmosis. I'm talking about endoosmosis happening here. So gradually what do we get to see that the whisking bag swells up, fills in water, takes in water. And progressively what will happen is the level of the water in the beaker will gradually start dropping off. So what has happened here? What has happened here? The water has started moving from the beaker to the bag. The water is moving from the beaker to the bag. What about the concentration of the solution in the bag? Increasing or decreasing? Concentration of the solution. I'm not talking about the level. The level is increasing, of course. What about the concentration? Concentration of solution in the bag? Decreasing. Concentration, it has become gradually dilute because the water is getting in. If it was 20% earlier, now it must be something around 10% because so much of water has come in. Following or not? So this is what we are talking about, and the same condition here also. And if I attach a glass tube here, then same like the thistle funnel, we can see that there is a rise in the water level or the level of the solution. So same is there in your book also. If you check that figure with whisking bag, check the figure. Same there. So with the whisking bag, they have attached one glass tube. So what will the glass tube help us in understanding? Whether the glass tube will help us in understanding whether the level of the solution is increasing or not. And will the level of the solution increase? Will the level of the solution increase or not? Tell me. Yes. The level of the solution will increase. Something like, if I talk about this whisking bag here, say suppose now I talk about this whisking bag here, and then I'm just inserting a glass tube. And there is the beaker, and the beaker has the water outside. So after the particular period of time, I may just get to see after some 3, 4, 5 hours, as the bag starts getting swollen, the level of the solution in the glass tube also rises. And will this progressively keep rising? Yes. Till when? Till the concentration. Now this is a semi-permeable membrane. Till the concentration here and here is the same, equilibrium is reached. So this is what we are talking about as a concept of osmosis. So this basic concepts of osmosis we have already talked about. What did we understand? With the cellophane, with the rubber sheet, with the muslin, with the whisking bag. Now, when I talk about the movement of the minerals, now something little bit when we talk about movement of the minerals and the water, absolutely different. Remember, when I have to talk about a cell, now I talk about a cell. This is a plant cell, and this is the cell sap, and the minerals and the molecules of the minerals. When I talk about where are the minerals or where are the different type of solutes more, soil or cell? In the cell, the molecules of the solutes are always found to be much higher. But then, does the cell actually take up these minerals from the soil? They do. They do. The answer is, even though the cell has so much of it, the cell still would love to take these minerals inside. But here it is a concentration, it is a low concentration area, no? And this is the high concentration area. I'm talking about minerals right now, not water. So the moment I talk about minerals, this is a low concentration area. I am talking about the soil. And the sap has got more of it. Then how is it possible that the cell will take it? Now the cell will take these minerals even though the cell has already a large amount of it. But for this, the cell will happily utilize energy molecules in the form of ATP. Now the cell will very much utilize energy molecules in the form of ATP, and this process by which the cells, the plant cells, are constantly taking in minerals from a region of low concentration to a region of high concentration. But with the utilization of ATP, this particular process is known as active transport. This particular process is known as active transport. So, can you tell me a difference now between active transport and diffusion? Diffusion is
there is no use of ATP.
There is no need of any diffusion or passive transport. The moment we are talking about diffusion or passive transport, there is no utilization of energy in the form of ATP. But the moment I talk about minerals, the moment I talk about solutes moving from soil to the cell is actually taking up minerals which already the cell has lots. But still the cell wants to keep it as surplus. The cell still wants to grab them because it is constantly utilized in the formation of the pigments and the hormones and the enzymes. But the cell will always love to utilize energy molecules to grab them because the movement is known to move against the concentration gradient. Come across certain terms. The movement of the ions here are against the concentration gradient. What does that mean? The movement is not from a region of high concentration to low concentration, but the movement is always from a region of low to high. If this is called a movement against concentration gradient, then for water molecules we should always say along the concentration gradient. Osmosis takes place along the concentration gradient. What does that mean? Along meaning from a region of high to low. Against meaning from a region of low to high. And if it is region low to high, then ATP molecules are involved. Take one question. Write two differences between passive transport and active transport. Passive transport and active transport. What happened? Make a column. Passive transport and active transport. Passive transport and active transport. Right under passive transport. Under passive transport, write down fast. Under passive transport, write the movement of passive transport, movement of molecules from its region of high concentration from its region of high concentration to its region of low concentration. Movement of molecules from its region of high concentration to its region of low concentration. Active transport, movement of molecules from its region of low concentration to its region of high concentration. Movement of molecules from its region of low concentration to its region of high concentration. Second point, passive transport. What did I say? Passive transport can also be understood as diffusion. Passive transport can also be understood as osmosis. Remember, these are all passive transport concepts. Under passive transport, there is no utilization of ATP. There is no utilization of ATP. There is no utilization of ATP. Active transport, energy in the form of ATP is utilized. Energy in the form of ATP is utilized. Energy in the form of ATP is utilized. Third, diffusion, passive transport, applicable for water, solute, gases. Applicable for water, meaning you can write solvent, solute as well as gases. Active transport, applicable for molecules of solute, minerals, etc. Applicable for molecules of solute, especially minerals, salts, ions, etc. So this is a basic difference between active transport, passive transport, and based on certain conditions inside the bracket, what you are asked, you will be always talking about that condition. The last concept that we complete here now is, we have been talking about active transport already. We go back to the concept of osmosis now to understand something different. Here, what I am telling you is, I told you already a situation something like this. No? Now, little bit of a modification and tell me what it should happen. In this case, the same thing that I am talking about. There is the water outside, and then the thistle funnel. The thistle funnel has a solution, and then now the mouth of the thistle funnel is fastened, closed, shut down with a cellophane or a parchment. So all these things exactly remain the same. Now, when I'm talking about that, this type of a concentration difference is already there. Now, this type of a concentration difference is already there. But apart from this, now what are we doing is, we apply certain amount of weight here on this. We apply on the surface of the solution. We apply certain amount of weight with a piston, and we apply certain amount of weight. So what are we doing here? We are taking a piston with a weight. We take a piston, and on the piston we keep some weight. Now tell me, what should happen? What happened?
She is asking time.
घर जाना है, भूख लगी है. So now that we are talking about this type of a condition, try understanding what is the difference. What is the difference? Just 2 more minutes. Now that we are talking about this, what will be the difference you feel? Water, solution, everything rests same. But now that there is the funnel, होली में वो पिचकारी खेलते हो ना? Exactly same type of a piston applied, and on top of that some weight is kept. A same type of a piston and a weight kept. Now tell me, should the movement of water happen same way from the beaker to the funnel? No. Why not? Because of the way there is a pressure which is generated on the inner side of the cellophane or the compartment, which is not allowing the water to enter. This particular pressure which is preventing osmosis happening, it is preventing osmosis, no? This particular pressure which is preventing osmosis happen is known as osmotic pressure. The definition of osmotic pressure, please take a note. What is the definition of osmotic pressure? The minimum pressure which prevents the movement of the solvent into a solution. Otherwise, if this was not there, this would have happened. Okay? So the minimum pressure which does not allow osmosis to happen, that is what is known as osmotic pressure. We'll continue again with this chapter the next class.
हो गया
दूसरा अरे ड्राइंग अगर मैम ने देख ली ना
सूर्य से क्या है ना
अरे तो इसका रेडी वर्जन देखो ना
guys, the
सूर्य
basic
snap
अरे हम सिंग
मोटिव
Is the Suez
Yeah,
Canal?
one
Suez Canal, or call it the Suez.
अच्छे से आया
थोड़ा फास्ट चले ना
मेरे को तो नहीं लगता आएगा मुझे बहुत टाइम लगेगा
क्या डाउट बनाता मैम को दिखाने के लिए
So, good, yeah, what's that?
मैम सुबह दी है डायग्राम बना रहे मैम 3-4 बार इसके देखे वो कहीं मार रहा था जल्दी चल भैया देख ना तुम मारेंगे जल्दी चल वो
इरेज कर दे क्या
जल्दी आए बंद कर दिया जल्दी चल
मेरे को लगा है
अरे पागल है
अरे भैया देखना ना सब सीधे अपने को है
नहीं भाई तेरे ऊपर आएगा पीछे कैमरा है
अपने ऊपर क्या
वही ना
मैं तुम लोग को बोलूंगा तुम लोगों ने बोला
क्या वो कैमरा है भाई कैमरा से छूटे
वो लोग बोलेंगे सुसाइड कर लेगा
वही ना
वो लोग
हम बोलेंगे खुद की मरवा देगा
साइड अलग है
अरे बस
अरे चल
प्रैक्टिकल बात करता हूँ
बाजू
हम लाइव स्ट्रीम चालू है क्या
हो गया
तेरे लिए ऊपर चढ़ना
हाँ मैंने कहा
यहाँ पर भी तुमने बनाया
इधर वो तो हल्का सा बना रहा है
चलो ना
चलो हमको तो नीचे चलो
उसको पता है
हाँ,
मैं गिर जाता हूँ